Ultra-purpose liquid chromatography is established technology for accurate analysis of IgG N-glycan due to its superior sensitivity, resolution speed, and its capability to provide branch-specific information of glycan species. This method is easy to use and has a number of different advantages: relatively low cost of equipment, high reproducibility, robust quantification, the ability to effectively separate glycan isomers. The measurement of protein glycolysis in plasma could be an attractive, minimally invasive biomarker and assist patient stratification for precision medicine. Our previous research found that the association between IgG in glycolysis and certain diseases, such as hypertension and biological disease in Chinese population. You know that the method of UPLC is used to test IgG N-glycans. Actually, it can test the N-glycans of protein, which is needed in a corresponding protein extraction plate. We believe that a method of UPLC can be applied to the field of protein post-translational modification. The overall experimental process is relatively simple, but the experimental process needs to be standardized. The entire experimental process takes 3 days to operate, and the experimental steps need to be memorized in order to master the experimental skills.
To prepare the samples, thaw the frozen plasma sample. Then centrifuge at 80 times G for 10 minutes. Leave the protein G monolithic plate at room temperature.